This is the screencast corresponding to May 30, and we'll be talking about hormone regulation, that is circadian regulation of hormone release. And just to get a sense of how dramatic, endocrine release is regulated by the circadian system, here's on the next slide an example from humans. Um, these are several hormones in humans that are regulated dramatically by the circadian system. In fact, there's not a single neuron, a single hormone that it's not regulated by the circadian system. If they're not directly regulated by the circadian system, they're typically regulated by sleep, by the sleep process, specifically by some sleep stages, and that means that in turn they're regulated by the circadian clock. But most of them are directly regulated by the circadian clock. And here are some examples. We have cortisol here. Let's see. And we said that cortisol is one of the glucocorticoids that in rodents we typically measure corticosterone. But in humans, we typically measure cortisol. You've probably heard of this hormone in the context of stress. It is kind of the typical stress hormone released by the adrenal gland. But in fact, cortisol has also a normal circadian rhythmicity in its release. Um, Tyrosine stimulating hormone. This is the hormone that stimulates your thyroid gland to um, to release tyrosine. Melatonin. We've talked many times about melatonin, that it's released in a circadian fashion. Um, Same for prolactin, a hormone that is essential to sustain pregnancy and also to sustain lactation. Um, and the same thing happens for growth hormones. Growth hormone is under sleep regulation. It's typically released during slow wave sleep, and that's why you see this big peak at the beginning of the night. So this is nighttime. And then below we have this sleep regulation: REM, non-REM, and REM sleep, or slow wave and REM sleep, just to for you to get a sense of when these hormones are released. What's really interesting is that although all these hormones, and most hormones, as I said, are circadian in essence in their release, what the timing, their phase of release is different. For instance, we see that growth hormones is released at the very beginning of the night, whereas cortisol is released at the beginning of the daytime. And in fact, cortisol is a hormone that prepares you to be ready for the day. It's kind of a signal in the body to prepare you for the daytime activity. The main point that I want to make with this slide is that, in fact, there's virtually no hormone that it's not released in a circadian fashion, and also that if you think about this as the normal pattern of release, in fact, if the abnormal, if cortisol would be intermediate levels all the time, or melatonin would be low or high or intermediate all the time. Any of these hormones, in order to be released in a normal physiological pattern, they need to cycle. And this kind of, in a way, completely challenges the notion of homeostasis. You've heard about this term in your physiology classes, even in introbiology, and the idea of homeostasis is that biological systems tend to maintain a constant um, internal medium? Well, there's nothing constant in any of these hormones. In fact, if you look at any of them, they're oscillating sometimes 20-fold, sometimes 30-fold. So there's a huge variation in the patterns on the pattern of release, and in fact, that's the normal pattern. And in fact, now knowing what we know about um, circadian rhythms, what we talk about is more predictive homeostasis. And what that means is that the way in which your body stays at equilibrium is not by staying the same all the time, but actually by oscillating and preparing you for the changes that are about to happen in the external environment. External and internal, because we've already learned that in fact there are many internal oscillations that need to be in synchrony with each other. So by now, and you probably are familiar with this concept, it is pretty clear that in fact hormones are regulated by the brain. But back in 1940s and 1950s, this was not completely settled. You could take an ovary and transplant it from one female into a female that had no ovaries, and that could reinstate the reproductive cycles. You could do the same with testes and many other glands. So the idea was that these glands were autonomously releasing hormones. However, there were some classic experiments first in rabbits with luteinizing hormone, and then in rodents. In this case, this experiment is done in rats. That showed that indeed the release of hormones was regulated by the brain. Although the hormones were being released by glands, these glands were in turn under brain control. And this is one example. What we have here is two groups of animals. And these animals, this is a paradigm that we use very much in reproductive biology. These are females, okay? And in this case, these females have no ovaries, so they are ovariectomized. So, or OVX. And essentially, the reason that you want to ovariectomize is that you want to remove the endogenous source of estrogen. So basically, there's no circulating estrogens in these females. If you actually inject, if you take ovariectomized females and you add exogenous estrogen or estradiol, which is typically abbreviated as E2, is one type of estrogen, and it's the most common estrogen released by the ovary in most vertebrates. So if you actually add constant estrogen to an ovariectomized female, interesting, what you get is that instead of getting that female to cycle normally, which would be what you expect in a human female, you expect one luteinizing hormone release or one luteinizing hormone peak every 28 days. In a rat or a mouse, it'll be the cycle lasts 4 to 5 days, so it's shorter. So one would think, well, if I replace the hormone that the ovary produces, maybe I will get the pituitary to release luteinizing hormone in a cyclic way, that is, in a 4 to 5 days or a 30-day in women, okay? But it turns out that doesn't happen. If you inject constant estrogen in a mammalian female, at least, what you will get is if you have successive days, and this is light, dark, and so on. So this is, let's say, the dark, the dark, the dark. what you will get, instead of getting one peak every 4 to 5 days or every 30 days in humans, what you will get is one daily peak of, yes, one, let's see, sorry. So you'll get a peak every day instead of a peak every 4 to 5 days, or a peak every day in women instead of a peak every 28 days. So basically, if this is your night and your days shown here, this would be luteinizing hormone levels. What you will get is in a rodent, you will get a peak at the end of the day, and then another peak at the end of the day, and another peak at the end of the day. So this was very surprising when this was first found. And people thought, well, turns out that there may be some daily signal that times this luteinizing hormone release. By the way, for those of you that have not seen or do not remember, you should have seen this. Your basic endocrinology, luteinizing hormone is the hormone that induces ovulation, and anytime you have a peak of luteinizing hormone, you will get ovulation. So in this female that has no ovaries, in which you put constant levels of estrogen all the time, you get a signal to ovulate that has a daily timing. That is, every single day you get a luteinizing hormone surge. Whereas under normal conditions, in a female that has ovaries, if it is a rat, you'd get this every fourth day. So when people saw this, they thought, well, this is kind of interesting. It looks like there's a daily signal controlling the release of hormones. Where's that signal coming from? And the first idea that this was coming from the brain came from experiments that stimulated the hypothalamus and led to the release of luteinizing hormone, to the increased ovulation. But in this experiment, what they did is they had two groups of females. They were all ovariectomized, and they also had two constant levels of estradiol or estrogen. And they have one group, which is shown here with black circles, and I'll draw this in red. So this group, they just put the constant levels of estrogen and let them be. And what they saw is there was one peak on day 1 and one peak on day 2. But then they had another group in which they added, and in this case, these females also got a saline injection. That is the control solution. In the other group, which is shown here with these squares, black squares, and starts here, they injected, instead of saline, they put barbiturates. In this case, the barbiturate was pentobarbital. And interestingly, when they put pentobarbital, they completely blocked the first day luteinizing hormone peak. In other words, if you look at this curve here, this is flat on day 1, and then on day 2, there's a peak, which is at the normal timing. So there were two interesting findings out of this experiment. The first one is that we knew by then that barbiturates acted on the brain. So that meant that you could put a drug, a sedative, a very strong sedative, that works at the brain level, and that would result in inhibition of the release of a hormone by the pituitary, luteinizing hormone. The second finding was that the injection of pentobarbital didn't block the surge for a few hours, and then the surge took place. In other words, phenobarbital was injected only here, right? At this time window. And the expectation was that when the phenobarbital was washed away, you'd have this peak and leave it delayed. But that didn't happen. The system, this luteinizing hormone peak, actually had to wait 24 hours in order for that luteinizing hormone surge to happen again. So again, this was showing that that 24-hour signal was coming from the brain, and that if you blocked it now, you didn't get the surge a couple of hours later when the drug was washed away, but you'd only get it 24 hours later, indicating that there was a 24-hour window of opportunity for the luteinizing hormone to be released. Um, so that kind of settled that not only there was a 24-hour signal, but there was also that that 24-hour signal was coming from the brain. And in fact, this is relevant not just for rats and mice, but also for women. I told you that in a rat, if you have a light-dark cycle, the peak of luteinizing hormone, which corresponds to the signal to ovulation, occurs at the end of the day. In humans, that peak occurs at the beginning of the day. And of course, this is every 4 days in rodents, and this is every 28 or whatever the length of the reproductive cycle of the specific woman that you're looking at occurs. But interestingly, the timing of this surge is different, which means that a rat will typically ovulate just before the beginning of the night, whereas a woman, and typically diurnal animals, will ovulate just before the beginning of the day. And the idea is that throughout evolution, this timing of ovulation was synchronized with the pattern of activity, because the idea is that your chances of getting pregnant are increased to maximum. So that is, you will have ovulation, and after that, you will start your activity where you will seek a mate and achieve pregnancy. Of course, nowadays in humans, this is highly cultural, but in principle, the rhythmicity of luteinizing hormone release is still maintained, and in most women, the luteinizing hormone surge occurs in the early morning. This is, of course, important because, in part, this timing is very important for in vitro fertilization. In fact, in the early morning, you're much more likely to induce ovulation, because that's when the signal comes out naturally. One experiment that somebody did a while ago was to look at another phenomenon that is directly related to the release of ovarian hormones, which is the rhythm of estrus. And what we mean by estrus in rodents is sexual receptivity. It turns out that in female rodents, there's a rhythm of sexual receptivity that coincides with the timing of ovulation. Interestingly, this rhythm is also present in humans. That is, sexual receptivity in the female increases at the time of ovulation, and again, that's in principle a mechanism selected to increase the chances of pregnancy. In rodents, as I said, and in this case, this is a female hamster, this rhythm of sexual receptivity is also synchronized with ovulation, and it takes place every 4 days. And what you have here in this hamster under constant conditions and this is a hamster in relatively bright light, I mean, not constant darkness. And what you see is that every time you have an asterisk, is when the female is shown to be receptive to the males. Interestingly, if you put this female in the presence of a male at any of these time, right, at any of these cycles, every fourth cycle, they're very sexually receptive. If you put them in any other day, in any other of the 3 days, they will reject the male and they will actually be extremely aggressive to the male. What you have here is an actogram with the stars indicating when the female was receptive. And interesting, what these researchers did is in this period here, which only lasted for 3 days, they gave this animal deuterium water. So this is water made with a nitrogen that is heavier. And interestingly, people knew that when you used the deuterium water for drinking, the period of the circadian rhythms would get longer. And you can see that very clear, that it went from a long period to a much longer period. And then when they resumed the regular water, the period went back to what it was. However, the point of this experiment was not to show that deuterium water had the ability to lengthen of the circadian cycle, but in fact that when you lengthen the circadian cycle, you also lengthen the separation between these dots, right, which indicated sexual receptivity. And the point of this experiment was to show that whatever was regulating the timing of sexual receptivity, it was also regulating with the same timing, but every fourth day, the timing of circadian rhythms. And the conclusion from this study was that estrus, or the rhythm of sexual receptivity, was always a 4 times multiple of the circadian period. So whether the female circadian period was 22 hours, 20, let's say that this is 25 hours or 27 hours, always every fourth cycle that female was sexually receptive. And this was very strong evidence that the same clock that regulated locomotor activity was regulating the luteinizing hormone surge and the whole reproductive response that was associated with the luteinizing hormone surge. If you So people started thinking, well, if that's the case, it may mean that the same clock that is regulating circadian rhythms is also regulating the release of hormones. And this was the first experiment to do SCN lesions. We saw one example of that, which showed that SCN lesions abolished locomotor activity rhythms. This is an example in which they show that SCN lesions abolish the release of glucocorticoids. In this case, it's corticosterone. These are rats. And essentially what you see here is if you do a sham operation, there's a peak of glucocorticoids at the beginning of the night. That's typically for rodents. And basically what it shows here is if you do a cut that is a knife cut rostral to the suprachiasmatic nucleus, you see that there's a decrease in release. But most importantly, if you lesion the suprachiasmatic nucleus, the rhythm of glucocorticoid release is gone. Okay, and you can forget now about the different types of cuts. But the point of this was to show that indeed, if you lesion the suprachiasmatic nucleus, the release of glucocorticoids was, the circadian release of glucocorticoids was gone. So these, together with the experiments that showed that the rhythms of activity were gone, were kind of the first evidence that the SCN could in fact contain a clock. However, you've already know that a lesion on itself doesn't really show you that um, the place that you lesion is the clock, right? It could be that the suprachiasmatic nucleus in this case is a link between the clock and the release of glucocorticoids. So more experiments had to be done in order to determine how is it that the suprachiasmatic nuclei, and this is schematically the suprachiasmatic nucleus with many neuronal clocks that you already know that each neuron in the SCN is a clock. We already knew by then quite well how the SCN regulated locomotor activity rhythms, but we really didn't know how the SCN could regulate the release of these hormones. So let's look at that evidence. So so you already know the Ralph experiment. This is again a classic experiment which showed that in fact locomotor activity rhythms in SCN lesioned animals were rescued, but interestingly they were rescued with the donor specific period, not with the host specific period, indicating that truly the suprachiasmatic nucleus contained a clock that regulated circadian rhythms of locomotor activity. So the question that you may think about is, well, can I do the same experiment and look at whether hormones are also the circadian release of hormones is also rescued by the same transplant? And that experiment has been done. So what was the outcome? And this is one study from Eric Bittman's lab. And essentially, in this case, they repeated the Ralph experiment. So they did SCN lesions, they transplanted the suprachiasmatic back to these SCN lesioned animals, confirmed that they all, that all the animals that they transplanted the SCN, the rhythms were rescued, the locomotor activity rhythms were rescued, and then measure hormones in these animals. And the question was, in these animals in which the locomotor activity rhythms had been rescued already, were the hormone release rhythms rescued? And here we have the result for corticosterone. This is an animal that received a sham lesion, so in other words, it went through the anesthesia, the whole surgery procedure, but the SCN was not lesioned. And these animals, this is several animals sacrificed at different times, have a beautiful rhythm of corticosterone release. However, in the animals in which you lesion the SCN, you transplanted the SCN, you rescue the locomotor activity rhythms. If you look at glucocorticoid release, the rhythms of glucocorticoids were flat. There was no rescue rhythmicity. Then they also looked at melatonin, and same thing, the rhythms of melatonin in the animals that you transplanted and you did rescue locomotor activity rhythms were gone. However, in the sham operated animals, there was a peak of melatonin release at night as you'd expect. And finally, in females that are ovarectomized and implanted or injected with estrogen, you see that again in that group, there's no rhythm whatsoever. However, in the group that was sham operated, in the control, there are two nice daily peaks of luteinizing hormone, LH. So the conclusion from this experiment was kind of surprising. This means that although suprachiasmatic nucleus transplants could rescue locomotor activity rhythms, they were unable to rescue the release of the circadian release of hormones. And there are two basic interpretations that you can make out of this experiment. One would be that maybe the suprachiasmatic nucleus was not the clock for hormone release, or maybe that in fact, in order to rescue endocrine rhythm, the release of hormones, what you needed was specific neural connections between the suprachiasmatic nucleus and specific targets in the brain. We saw, in fact, from Ray Silver's transplant experiment that you could actually wrap up the suprachiasmatic nucleus in a polymer, and that would still be able to rescue locomotor activity rhythms. In this case, none of these transplants is wrapped up in the polymer, but still they cannot rescue endocrine rhythms. So one interpretation of these results is that maybe in order to sustain rhythms of hormonal release, you need to, in fact, make very specific synaptic connections between neurons in the SCN and neurons that are involved in the release of hormones. And in fact, when people started thinking about this idea, they thought, well, what could be the hormone, the neurons that receive direct input from the SCN to regulate endocrine release? And as you probably know, the way in which most hormones that are circulating in our body, or most of the ones that you know at least, are regulated is through the hypothalamus pituitary axis. You've already talked about this in Biology 220, but the whole idea is that there are neurons in the hypothalamus that project to the portal system of the hypophysis or anterior hypophysis or pituitary, which are synonyms. And here, these neurons release hypothalamic releasing hormones that in turn will get into cells and induce other cells that are not neurons and induce the release of pituitary hormones. And going back to our example of luteinizing hormone, if we're talking about luteinizing hormone as an example, the neurons in the hypothalamus that will induce the release of luteinizing hormone are gonadotropin releasing hormone, GnRH. So this hormone, GnRH, will be released into the pituitary, and in turn, specific cells that have receptors for GnRH will release luteinizing hormone, or LH. So this is gonadotropin releasing hormone, and this is luteinizing hormone. So that's just one example. So the first thought was, well, because the suprachiasmatic, these cells, GnRH, and the same is true for tyrosine-releasing hormone. There's another set of neurons that make tyrotropin-releasing hormone, and for growth hormone that make growth-releasing hormone, and for adenocorticotropin hormone, which is gonadotropin-releasing hormone. So there are many different neurons that will specifically induce the release of a specific hormone in the pituitary. So because these neurons are in the hypothalamus, people thought, well, maybe the suprachiasmatic nucleus neurons have direct input into these neurons, which in turn are responsible for the circadian, for the release of hormones, right? and it's this SCN input that makes this release circadian. So when I started doing my PhD back many years ago, this is one of the questions: can the SCN itself, can neurons or do neurons of the suprachiasmatic nucleus directly talk to neurons that are making hypothalamus releasing hormones like gonadotropin? And this is shown in this experiment. So basically, what we did in this experiment is we had in the brain, in the suprachiasmatic nuclei of females, we injected an anterograde tracer. So essentially, as you already know, I mentioned this several times, an anterograde tracer will be a molecule, a substance that will be absorbed by the cell bodies of the neurons in the suprachiasmatic nucleus. Imagine that this is one neuron, and it will travel down the axon, and then because that anterograde tracer is not made by the hamster, you can actually label it and see where the neurons from the site at which you injected the anterograde tracers project to. And what we did in this study is we injected that anterograde tracer in the SCN, the suprachiasmatic nucleus, and then stained, labeled that anterograde tracer, which is labeled here in black. These fibers that are shown here are axonal fibers that are coming from the SCN, because I know that I injected this anterograde tracer in the SCN itself. And then this brown cell, large cell, is a cell that makes GnRH. In fact, LHRH in this case and GnRH are synonyms. It's luteinizing hormone releasing hormone or gonadotropin releasing hormone. But the point is that you can see many of these black fibers arriving to these GnRH neurons, indicating that fibers from neurons that are coming from the suprachiasmatic nucleus have the ability to directly communicate with GnRH neurons. And in turn, these GnRH neurons are the ones that will regulate luteinizing hormone release. Efferents, by the way, means axons projecting out of, so this SCN efferent means SCN axonal projections. And one thing that it's important because of what comes afterwards is that these projections are what we call predominantly ipsilateral, which means that the right-sided SCN projects mainly to GnRH neurons that are on the right side of the brain, whereas the left-sided SCN projects mainly to GnRH neurons that are on the left side of the brain. So there's sort of a lateralization a bias in the way in which each SCN projects to. Each SCN would project to the same side. Okay? So these confirm that indeed the suprachiasmatic nucleus had very specific projections with neurons that were essential for the release of pituitary hormones. However, these still didn't show that, in fact, those fibers were critical for that indeed the SCN had to make synaptic contacts in order to drive those rhythms of luteinizing hormone release. Remember, the main question is why did the transplant experiments not work for endocrine rhythms, but they did work for locomotor activity rhythms. And one experiment that I did several years later kind of answer that to some extent, that question. So as you already know, if you study the expression of clock genes, in this case it's period one in the suprachiasmatic nucleus. Typically, PER1 peaks during the light phase or the subjective day, and is not expressed during the subjective night or during the dark phase. So basically, this is a section of a hamster brain, and in this case, it was labeled with a probe that labels a messenger RNA for period one. And you see that there's a lot of expression in the 2-SCN at the ventral part of the brain in the middle of the day, but if you do the same experiment in another animal in the middle of the dark, what you see is that there's no expression whatsoever. So, and we've already seen from other studies, from when we talked about molecular biology, that there's an oscillation in period expression so that it peaks during the daytime and it troughs during the nighttime, right? Okay. Expression of PER1, where you have both SCN very nicely labeled, okay? So there was one specific behavior in the hamster called splitting, which was very peculiar. And this was that hamster, when they're put in constant light conditions, this is LL, so instead of putting them in constant darkness, you put them in constant light all the time. And what you see is that most of the animals will do this very unusual behavior where you go from a consolidated circadian bout of activity where the period has around 24 hours. It's longer than 24, but close to 24. Suddenly, the animals split, and you get two bouts of activity within the same animal. So these animals switch from a close to 24-hour rhythm to a close to 12-hour rhythm. Okay? So this is, quite remarkable, and people did not know why this happened. And what they decided to do is to see what was the pattern of gene expression for period one, period two, and period three. But the most important ones are period one and period two. This PER3 is not a clock gene in rodents. What happened with the pattern of expression in these animals that have undergone splitting? And what we saw was quite remarkable. At some times of the cycle, if you sacrifice an animal, what happened was that one side of the brain, that is the right side, for instance, in this case, of the SCN had high levels of PER1, whereas the left side had no levels of PER1 whatsoever. And the same happened for PER2. So it was almost like in the same animal, at any given point, one suprachiasmatic nucleus was showing very high levels of expression of PER, which corresponded to daytime, and the other side of the SCN, the other SCN, was showing low levels of expression, which corresponded to the night. Compare this pattern. Let me erase this. Compare this pattern of expression of PER1, where you have only one SCN labeled, to this. So that was very surprising, and actually we did many experiments to show that what happened in animals that were not split, the two suprachiasmatic nucleus were oscillating in sync with each other, so that when the left side SCN was showing high levels of period one, the right side of the SCN was also showing high levels of period one, and vice versa, when they were showing both low levels of period one. However, in the split hamster, what happened was that they somehow had come out of phase 12 hours or 180 degrees, so that when the right side SCN was showing daytime levels of period one, the left-sided, the right-sided SCN was showing low levels corresponding to the nighttime, and then 12 hours later, the situation reversed. The left-sided SCN was showing low levels corresponding to nighttime, whereas the right-sided was showing high levels corresponding to daytime. So what this means is that at any given point, one half of the SCN, the left half, was telling the animal it is daytime now, whereas the other half of the brain had a clock that was 12 hours apart and was telling the animal it is nighttime now. So this became kind of a useful system because now we could have one half of the brain in daytime mode and the other half of the brain in nighttime mode. And remember, I told you that the right-sided SCN projected to neurons that had GnRH that were on the right, whereas the left side SCN projected to GnRH neurons that were on the left. So the question was now, okay, if indeed these projections that go from the SCN to each side of GnRH neurons are functional projections, what we should see is that the GnRH neurons on the right side of the brain at any given point would show signals that corresponded to the daytime, whereas the left side neurons would show signals that corresponded to the nighttime. So there's one way of looking at that, and that is, you can forget about this, but the main point is, what happens to the output of these neurons. So one thing that we knew is that female hamsters that were ovariectomized and implanted with estrogen, with estradiol, that split had two surges of luteinizing hormone. So instead of having the normal one surge, one every 24 hours, these females were getting a signal from the brain that was telling them to ovulate twice a day instead of once a day. Okay? Again, this was also evidence that the same clock that was timing locomotor activity was timing the release of luteinizing hormone. Okay? So the hypothesis here, when we knew about this, and we knew that the two SCN in these animals are left and right in opposite phases, the hypothesis was that this bout of activity came from, say, the left side SCN, whereas this bout of activity came from the right side SCN, and in turn, this luteinizing hormone surge came from the left side SCN, and this luteinizing hormone surge came from the right side SCN. So how could we look at that? Well, it turns out that there's one marker of neuronal activity that you've already heard about, which is CFOS, which you can also use to know which part of the SCN is active, this is daytime, and which part is not active, which is in nighttime. In this case, we label with an antibody against CFOS, the protein. Okay? And this is an animal that was split and was sacrificed here. But in this case, these are females that were ovariectomized and implanted with estradiol. And then the question was, in these animals in which they're splitting, and one SCN is telling the animal is daytime, and the other, the SCN, is telling the animal is nighttime, are the neurons that have GnRH that are on either side of the brain also getting this signal, daytime and nighttime? And we can also do that with CFOS. It turns out that at the time of ovulation, on the daytime, that is the end of the day, remember, in hamsters and rats, ovulation occurred here at the end of the day, and that corresponding with the luteinizing hormone surge. If you actually look at CFOS in these animals, in GnRH cells, what you will see is that most GnRH cells at this time will also have CFOS, meaning that they are highly activated. So what we decided to do is to look in these animals that are in splitting, that is, the animals that have one side of a brain that says nighttime and one side of a brain that says daytime, to look at whether the GnRH cells on either side of a brain were also activated and not activated. And this is what's shown here. So basically, this is one example from one animal within many animals, obviously. The side in this case was the right side that was on daytime. You can see that all these blue neurons, which are GnRH, blue means GnRH in this case, have brown nuclei, okay, here, and brown in this case, and here, means CFOS. So in other words, the cells that were on the daytime side of the brain were activated as you'd expect in a female that is about to ovulate. However, on the same female, if you look at the neurons that were on the left side of the brain, what you'd see is none of the GnRH cells had brown nuclei. So it did have some brown nuclei, but they were not inside those cells, indicating that the GnRH neurons that were on the left side of the brain, which in this case was the nighttime side of the brain, were not activated. So this was in a way a remarkable result because it was showing that, in fact, not only the suprachiasmatic nucleus was split into day and night, but also the rest of the brain that was receiving input, remember, lateralized input, ipsilateral input from the 2-SCN, was also in daytime and nighttime. So in a way, this female had half of the brain telling it you should be ovulating now, and the other half telling it you shouldn't be ovulating now, because one side was at the right time of the day to ovulate, and the other side was at the opposite, 12 hours apart, time of the day when they shouldn't be ovulating. So one thing that has emerged in the last few years is that, as you already know, we have peripheral oscillators all over the body, and the question is, well, do glands that release hormones also have clocks? And the answer is yes. In the same way that the kidney has a clock, the liver has a clock, the adrenal gland has a clock, the ovary has a clock, the testes, some cells also have clocks, and so on. So the question is, are those clocks able to modulate hormonal release even in the absence of brain circadian input? And as we'll see next class, the answer is yes. But I'm just going to show you first what is the evidence that in fact some of these peripheral glands have also clocks of their own. In this case, these are isolated GnRH cells. In fact, these are GnRH cells that are part of a cell line that came from a woman with a tumor, and essentially they make GnRH. They make a lot of GnRH, and you can actually culture them. And if you give them a serum shock in the same way that we saw for fibroblasts, you can see that you can induce very beautiful rhythms of period 1 and very beautiful rhythms of period 2, okay? Indicating that these GnRH cells have themselves clocks that are, at least able to generate rhythms of clock gene expression. And this is from another study, the U study, which shows that in fact there's a very nice 24-hour rhythm of luciferase. This is period 2 luciferase. Again, in this case, what you did is you took a mouse that had the promoter of period 2 driving luciferase. You took the pituitary, you put it in vitro, and you see that there's a very nice rhythm of bioluminescence. Okay, so and the question here is, well, if generate cells themselves and the pituitary itself is able to generate circadian rhythms, why do we need the SCN? Why do we need the brain to coordinate all these rhythms? And we'll leave it at that for the screencast, and we'll continue with the evidence that indeed um, these clocks also may have a role in controlling the circadian regulation of hormone release. So that's it for this screencast, and I'll see you on Tuesday.